So Liz, there's a lot of paths people can go on life. What's the path that brought you into fungi?
Well, it was just dumb luck, honestly. I got a degree in biology from Indiana University and wasn't really applying it in any way. And so I was like, I need to get a job in biology. Looked at all the random jobs in the bio posting, and there was one in a lab, and they studied arbuscular mycorrhizal fungi. And so I took this job as a 22-year-old and I guess fell in love with fungi, and I've been working with them ever since.
What do you think are some of the biggest misconceptions people have about fungi?
Hmm. Well, speaking of the type of fungi that I work with, specifically arbuscular mycorrhizal fungi. So when you think about mycorrhizal fungi, people are often thinking about this huge mushroom that's making all these connections in the soil, and they're talking to trees and they're communicating back and forth. And well, first of all, the fungi I work with don't make a mushroom at all. They're entirely subterranean, below ground microorganisms. So that's out the door already, what you're thinking of. But also, the way that these communicate with trees or possibly communicate with each other is definitely still a thing of debate in science.
We're going to go down that rabbit hole a little bit
Sure.
into theories.
Mhm. Mhm.
What are some proposed ideas of how this communication happens?
Yeah, so this is definitely not my realm, but I can summarize what I know, just being adjacent to this realm. But so the thought is that a fungus can be an intermediate between, let's say, two trees, because they share similar space in the soil, and a lot of fungi are not super particular who they associate with. Some of the ectomycorrhizal fungi, the kind that make mushrooms, are more sensitive to tree species, for example. But let's say we have two trees, the same species here and here. I could show you on my wall. They could connect below ground with the mycorrhizal fungi, and that fungi could and essentially allow those trees to talk to each other, communicating some sort of signal. This is a thing, this mother tree concept is something that is become very popular in science and is only partially supported by some science. Yeah.
Is there, if we go into more into your realm,
Yeah.
AMF,
Mhm.
is there communication happening between those and the plant? Is it just chemical? Because people who've listened to the podcast, they know, okay, AMF, there's a trade happening. We're
Yeah.
trading sugars for
For
stuff.
phosphorus. Yeah,
Yeah.
most of the time, but many things. Yeah, there is some selectivity going both ways. So we've done some studies where we take a plant and put its roots in two different pots. And in pot A, we have a fungus that's, let's say, amazing at acquiring phosphorus. And in pot B, we have a fungus that's not. And the plant can tell what fungi is giving it more phosphorus and can then allocate more roots to that fungus. So in that way, it can use space to segregate where it's giving its sugars to feed which fungi. So they can definitely be specific. And the reverse is also true. Some plants give out more carbon than others, and fungi require carbon to grow. They can't fix their own carbon. They can only be fed by these plants. And so some plants are better at feeding the fungi than others.
Are roots a direct link to fungal relationships? So if you see abundant, dense root mass, does that mean more fungi in the soil for
I would
trades?
say this is also a misconception I see a lot in the, especially in the cannabis world. For some reason, they show these big, beautiful root systems, tons of fine roots. What I see often, especially with these prairie plants, that when they have a really happy fungal community that's doing the work, that's their beneficial fungal community, they actually have less roots. So they can devote more energy to above ground resources and have fewer fine roots because the fungi are essentially acting like roots for them. The fungi are out in the soil, farming the resources and giving it to those coarse roots. And then the plant can actually allocate a lot less energy to fine roots. Yeah, so this is something I've seen time and time again.
So to bring people up to speed, maybe if they haven't listened,
Sure.
for arbuscular mycorrhizal fungi, how would you define that to the layperson?
That is tricky. They're very complicated little things. Where even the species concepts for AM fungi are they're all over the place. But essentially, what they are, they're plant simian. If I was going to talk to a layperson, I would say it's a soil microbe that can help plants. That's the easiest way to think about it. But they don't always help plants. Some are not very beneficial at all, for example. So there's lots of caveats to everything in ecology.
Can they survive on their own without plants?
They cannot. So they have this thing called a spore. And if you've looked at my social media or anything, I'm always posting pictures of spores. There's these little bright color circles. They're often yellow or red or somewhere in between. But the spore is sort of like their seed. So it's their dormant structure and their reproductive structure as well. And these spores can last for, in some cases, a couple of years. But they're not growing. They're just sitting there. So they can survive in their seed-like form, aka the spore, for a while. But they can't grow at all. They require a plant to feed them carbon continuously to grow.
So spores in the soil, dormant. Plant was there. There was a AMF community thriving. Plant's gone for whatever reason now. Spores are left. A new plant comes in. What's the trigger, the biological trigger to take that spore and say, oh, something's here. Like I need to non-spore.
That is the question, and something that the commercial industry of mycorrhizal inoculant production is interested in too. Like, what is dormancy and how to break dormancy? So some of the fungi, as soon as they're wet, they start to grow. They start to germinate. They start to creep out and look for a root. And if they don't find one, they die. Other species or other isolates might not necessarily grow right when they're wet, but they can send some sort of carbohydrate or sugar signal and then start to grow.
Is a spore similar to a seed in the sense of there's some carbohydrate stored energy that lets that fungi send out some hyphae for a certain distance, but eventually that food source is depleted, so it has to make that connection before it's gone?
Exactly, yeah. And these are microorganisms, right? So a spore it's a single cell, but it's one of the biggest single cells on the world. You can see it with your human eye, this single cell. So some of them can be quite large. Like the tip of a pen, around 500 micrometers, you can see these things. So they're packed with a bunch of nuclei, tons of genetic information, and a bunch of fat, lipids. And yes, they use this to start to grow.
So hyphae's out there wiggling its way through cracks in the soil. It comes upon a root. Do we know what then happens to initiate that exchange? Is it like, can the hyphae sense a chemical coming off the root? Can the root sense the fungi? What happens there that they don't just meet as two passersby and you'll just glance off of each other?
I think it is pretty random, right? But there is some chemical signaling, and that will allow the fungi to come in contact with the root and form the initial colonization structure inside of the root, which is called the apophysium. And so the plant will allow the fungus to do this. And there's been some work shown even in non-mycorrhizal plants like kale, for example. The brassicas are a type that don't associate with this fungi, but there has been some evidence that the fungi can form these initial structures and colonize the roots, but the colonization is very short term. The plant doesn't make space for this structure called the arbuscule, which is of the name Arbuscular Mycorrhizal Fungi, and that's where that nutrient exchange happens. So for some reason, in some plants, they have evolved to not have this structure and this association with these fungi, which is atypical because it's around 85% of plant families associate with arbuscular mycorrhizal fungi. And they've been around since basically the plants left the ocean, 465 million years estimated. So all that time as plants were crawling out of the ocean and exploring land, fungi were right there with them.
Are you familiar, or do you know a lot on the evolutionary side of things of the plant?
Only a little bit. I'm more ecology,
Yeah.
but a little bit.
Yeah, I mean, the thought is, right, the fungi, the hyphae are much thinner and longer than the root hairs, so they can get into more different areas. Why wouldn't the initial plants just have more and thinner roots? Why
just a ton of roots?
Yeah.
Yeah, good question. And, sometimes it could have been in a situation where fungi weren't around. Maybe plants would have went that route and they would just have gelatinous blobs out in the soil picking up nutrients. So who knows why fungi are? Sometimes nature just finds a thing and goes with it.
Okay. What about, so with this long, I mean, geologically long relationship between these two entities, the fungi comes in contact with the root. How does the plant become aware or enable it to colonize it? It
Mhm.
is does the fungi send out a signal that says doing it? Because I'm thinking, hey, I'm a nefarious one, right? I don't want to be a symbiote. I don't want to partner
Yeah,
with you. I want to take you over. I'm going to mimic this thing. You're going to let me colonize you, and then suddenly I'm on board the ship, and it's mine now.
it can be good or bad. Yeah. So I'm not an expert in this field, but I do know that fungi can trigger the jasmonic acid pathway in plants, which is sort of the defensive pathway. So the plant will recognize this thing as a threat, right? It's something coming into your body. So it can trigger that pathway, even though it ends up being a beneficial interaction. But sometimes triggering that pathway can also be a benefit to the plant, can prime it for other actual pathogens that or herbivores that it might encounter along the way.
And then when that connection is made, earlier you alluded to one of the main trades is carbon for phosphorus. Is phosphorus the main thing that a lot of the AMF are supplying?
Yeah, the AMF are phosphorus specialists for sure. And they're sensitive to the levels of phosphorus in the soil, and they're excellent at acquiring inorganic phosphorus, especially. But they can acquire a bunch of things, including nitrogen. There's been some work looking at water, calcium, and other micronutrients. So all these things in the soil, the fungi are interacting with, and they can acquire them and transplant them to their plant host, as long as they're getting fed well.
For people listening to this involved in gardening or ag, how does the relationship function in, say, a soil that has been fertilized with phosphorus versus and it's high versus one that's just more naturally low?
Sure. Well, I would say in the lower phosphorus environments, the fungi are essential almost, versus in many of our post ag landscapes and the landscapes that we're fertilizing constantly, that can actually lead to a situation where the fungi essentially become functionally redundant, depending on the plant. So if that plant has a lot of fine roots and also a lot of phosphorus, it might do actually okay without a fungal partner. And some plants, let's say onions, we've all picked up an onion and it has those little rootlets, they're very coarse roots. So onions don't have a ton of roots. No matter the phosphorus situation, they're going to be associating with mycorrhizal fungi because they just don't have the fine roots to even interact with enough phosphorus to support that plant. So it depends on the plant for sure. It actually takes quite a bit of phosphorus to make that relationship redundant, though, for most plant species. You have to fertilize a ton.
So if somebody's looking at supplementing their growing with an AMF inoculant, if we look at the benefits to the plant as a pie chart, is the biggest slice of that pie AM or phosphorus in terms of
I'd
the
say,
benefit?
yeah, the biggest benefit in most situations is going to be phosphorus. Yes.
Yeah, I was looking through some of the studies that you've worked on in the past, and one was the one you did looking at the plant community response to a 15-year long term of nitrogen and phosphorus enrichment in a grassland system. We found nitrogen and phosphorus enrichment resulted in plant community diversity decrease and composition change. What
It
can you talk?
also can, yeah, alters the fungi over time.
Yeah, can you talk a little bit
Exactly.
about that and how that looks in the real world? So what was happening on that land that you guys looked at?
Sure. So this was a study at the Kansas Biological Survey, which is where I work at the University of Kansas. And yeah, I mean, this is a really, really long-term phosphorus and nitrogen addition study. So many, many, many decades. I think now it's many decades. And the data we looked at, I think, was the 18th year or something, as you mentioned. But so, right, as you're continuously supplying this increased nutrients to plants, it can change the community composition in a couple of ways. So, for instance, some non-native plants and some weedy plants are the ones that tend to be more fine-rooted. So in a situation where there's really easy to access nutrients because of fertilization, these plants become more dominant. Versus in the unfertilized prairie, where nutrients are really low because they're managed. They already have pretty low nutrient, but they're managed via burning, which can remove additional nutrient accumulation. The plants that excel in that environment are the ones that can tolerate lower nitrogen and phosphorus. And they also tend to be the ones that are the more reliant on the mycorrhizal fungi too.
So you have weedy plants, more roots, fine roots. They can use those roots to go pick up all the abundance of nutrients that have been added to the soil. So why in ag systems you see a lot of those weedy plants show up.
Yes. Mm-hmm.
And at the same time, if they're doing all that work on their own, they don't need that relationship as much with the AMF.
For the most part, yeah. So they're not feeding the fungi as much and they're not supporting them. And that those fungal communities can shift in terms of what fungi can survive not being fed as much. It tends to be the smaller, weedier species that don't grow as much hyphae and are less beneficial. Those are the ones that can survive poor quality host plants, basically, parents that don't care about them at all. And so these fungi are proliferating. So it's a shift in communities and also a decline in some of those beneficial species over time.
So if we look at Kansas, if there was a native grassland that was there, and I don't know if that still exists,
Yeah.
but there's some microbial community underground functioning with the roots. If we compared that to somebody took out that grassland a long time ago, they started farming, and they walked away, and they're not farming anymore, and a bunch of weeds came in, and it's covered, there's plants growing, but it's not the native grassland that was there. It's weedy species, so it looks like, hey, this is abundant, there's plants all over it, it's thriving. But if you looked at that native grassland versus the recovering plot covered in weeds, and we looked below surface at the microbial community, what differences would there be?
Yeah, so these two environments are where I spend a lot of time comparing fungal communities across what I call old growth grasslands. So in Kansas, we actually have a ton of them. So grasslands basically have been around since the last glaciation throughout much of the Midwest. And basically from Kansas all the way to Indiana, from Canada pretty much to Mexico would have been this grassland ecosystem called the tallgrass prairie. And Kansas has some of the biggest patches of remaining tallgrass prairie of any state in the US. And I call these prairies old growth prairies or remnant prairies, meaning that they have not been disturbed. They haven't went through tillage and they haven't been overgrazed. They're just really nice prairies. They are naturally grazed by bison in the past and now with cattle at low density if it's a nice healthy prairie. And so the microbial communities in these spaces are amazing. And that's because the plant hosts for those fungi are amazing. So some of these prairie species can live more than a century. So they're sitting in the soil for decades and decades and decades and interacting with the fungal community in that same soil for decades and decades and decades. And so they have trained the fungi to be good partners for them because they're going to be there. And so these communities are often packed with super beneficial old growth fungi as well as old growth prairie plants. And that's in contrast to some of these more disturbed sites in the restorations areas that I've worked with in our post-agricultural landscapes. Basically anything you do that has disturbed the soil will disturb those mycorrhizal communities. Then the plants that reestablish tend to be the junkier, weedier plants. And they're also not very good at repairing the microbial communities. And so a shift in fungi based on the trait of land management can result in a shift in the above ground plant community. We've seen this time and time again.
Yeah, I know you've done some studies on that too. So if you had this weedy area and you wanted to get some tall grass prairie into it, is one of the paths to succession there inoculating it with AMF to enable those species to thrive over the weedy species that are there?
Yeah, so there's a couple paths in restoration. Managing the land in some way to reduce the nutrient accumulation from those weedy plants or from the ag history is important. So in Kansas, we often burn the landscape. I know that's not possible in all prairies everywhere. And so in those situations, they would hay it, mow it, and take that vegetation off. So the soil is becoming more and more nutrient limited, and that would support a community of prairie plants because they can function, and they do function very well in that environment. So that's one method, and it works. But so the bulk of my research is looking at what we call microbial transplants. And so what we do is we would go to those old growth prairies, let's say in that pocket of Kansas that has an amazing prairie, collect those microbes and take that whole soil into a new prairie environment to help the plants that require those microbes establish in that new environment. And so we do this with whole soil transplants, or in many cases, I just culture the mycorrhizae from that soil and move that into the new restoration.
What's the rate of application of something like that? This is obviously large acreage. Is it something
Sure.
as simple as spreading dust over a large area? So a really small volume of stuff goes an amazingly long way.
Yeah, so I've done a couple studies looking at inoculation density and also application method. So it depends how you distribute the inocula, how much you need. So these microorganisms are subterranean, right? So they spend their entire life cycle below ground. So finding some way to distribute that inocula below the soil surface is key. So if you were to just dust this along the surface of your plants or something, it would probably die very quickly because it hates the sun. It's not an organism that lives in the sun, it lives in the soil. So if you can find a way to inoculate your mycorrhizae below ground, that is key. And in terms of how much is required, that is a question we are still answering. So I've done a couple of these inoculation density trials, and it seems to be right around 32 kilograms per hectare is where we start to see some results, which is, that's many, many pounds per acre.
So in thinking of this and kind of putting it all together, if you want to ruin a prairie, start fertilizing it.
Yeah.
Then those plants don't need the fungal relationship as much because, hey, it's like you're just giving me this stuff. I don't have to work as hard for it. Why form a trade when you're just giving it to me? The fungi die off. And then when that fertilization goes away, those plants that are there can no longer survive without that relationship.
Yeah, exactly. And they're very dispersal limited, being subterranean organisms. They can move on worms and voles and a little bit in the wind, but they're not something like a dandelion seed that's going to float around and colonize the next door neighbor's prairie, for example. And so, yeah, once they're really disturbed, it's a major problem.
How much tillage is too much when we look at disturbing these communities? I'm thinking agriculture here.
Yeah. Well, ag is, tilling is, hot debate, I should say. Many people still do it, and many people are adamantly against it for many reasons. I think it's becoming more and more popular around here, at least, to do no-till agriculture. And I live in, row crop agricultural heartland here in Kansas, and where I'm from in Indiana. So it's encouraging to see less tillage going on, but that often is accompanied with more spraying of chemicals like glyphosate. So that's the downside to that, right? But in terms of how it affects fungal communities, it really depends on the timing of the tillage. So the fungi have this natural cycle where they are fed by plants, and when they're being fed by plants, they're often in their hyphal form, exploring the soil, getting in all those pockets and finding all that phosphorus. When at the end of the season, when the plant begins to senesce naturally or die in the case of annual row crops, that sends a signal to the fungi like, hey, they're not feeding us anymore. We need to prepare. So they'll go into their spore form. And the spore form is more resistant to things such as tillage. So if you till in the winter when the fungi are out of their hyphal form and in their reproductive state, their spores, they are more likely to be able to survive the next year. First is if you are going to till when the fungi are actively growing, some of these species don't reproduce very well from the hyphal chunks that are tilled up. And so those fungi would decline and ultimately die.
when we look at tillage, one of the factors or one of the variables in tillage is depth. Where in the soil profile are these microbial communities located?
Yeah, so much of the action is in the top foot of soil, no matter what microorganisms you're thinking about, in the grasslands and for most crop species. Some of the trees, there's a lot more fungi going deeper with those deeper roots for sure. That's more of the ectomycorrhizal fungi. But a lot of the fungal biomass and the fungal action and that acquiring of phosphorus and trading for carbon is happening in the 0 to 10 centimeter range. So it's towards the soil surface.
Yeah, so 10 centimeters, 4 inches, so really
Mm-hmm.
close.
Yeah.
Is it fair to say the shallower the tillage, the better?
Yeah, and I try not to till my garden ever, even though I throw my MycoBloom trash, we'll get to MycoBloom later,
Sure.
into my garden. So I'm constantly inoculating it, but I try not to till both because it also seems to release more weed seeds over time. So it just seems like a challenge, all this tilling, to what end? And I don't know the exact number on the number of tons of soil that are blowing in the wind across Kansas, but my car is covered in dust every day. And I'm sure you've heard of the Great Dust Bowl, right? This
Yeah.
tillage, all this wind erosion is a massive problem.
Yeah, because a lot of the farmers we work with, they're growing annuals
Mm-hmm.
and they're terminating them. So it's not this crop has run its life and it's as it starts to die off, the fungi sense that and they go. It's like, hey, I'm growing lettuce today. It's perfectly fine, but I'm harvesting it today. And it's just like, surprise, anybody in the
Mm-hmm.
soil, this plant is now gone. If you were to do that, so just, mid growth, just harvest the crop's gone. So those fungi are no longer getting that relationship, anything from that relationship, because the above surface is gone. And you went in and tilled there, you're probably doing some damage, right? Because it's still in that hyphae state and you're
In the
not,
hyphal stage. Yeah,
yeah.
they make spores kind of continuously, but they'll do the bulk of it when they're trying to senesce. Yeah.
How quickly does that period happen of spores like for senescing? So if a corn plant is dying, is it like, oh, I can detect it's dying and spores start happening over the course of days, or is it more like weeks or months?
Yeah, it happens pretty quickly. I would say weeks. So when we're growing our cultures, we kill our plants over about 2 weeks, and from it can go from hyphal stage there to just loaded with spores. So it can happen pretty quickly. These things can make spores. Exactly. And yeah, I don't know where I was going with the other end. Lost
Well,
it.
yeah, I've talked to people in the past. What are your thoughts on this of certain microbes pair better with certain crops, hence no crop rotation? So if you always grew, say, tomatoes, you always grew tomatoes in this area, the microbes in that area would favor tomatoes. And so you'd get a robust community for that crop. But then let's say you did crop rotation, which a lot of people advise, and you bring in onions, which you referenced before. Now it's like, oh, everything here was set up for tomatoes. Now onions are in place and it's a new species relationship. What are your thoughts on that whole theory of
Mhm.
leaving one crop in place to make a more robust microbial community, or it doesn't matter, you can rotate, it's all going to work out?
Yeah, I mean, so I study mycorrhizal fungi pretty much exclusively, but I come from a lab whose entire background is in plant soil feedback. So they think more broadly about the whole soil community. And for many of these lands that might only plant one crop over and over and over again, what they tend to see is a decline in biomass, not necessarily because of the AM fungal communities changing, but it's thought more of pathogen accumulation of pathogens that are specialists on those crops. So that's one of the main reasons that people do rotate things like corn and soy every other year to avoid those pathogens, which can overwhelm the benefits of any microbial community that accumulate depending on the kind of pathogen and what it might do to those plant hosts.
So rotation's probably better than,
Rotation's generally better. Yeah.
yeah.
Another thing, if you think about diversity in a space, that also can eliminate this high pathogen accumulation. So if you think about a prairie grassland, these plants live for 100 years, some of them, in that same exact soil. And why pathogens don't necessarily overwhelm that plant is because of all this diversity that's happening in the soil and the complex interactions of all those different biota. So there has been some research looking at diversification in crop plantings. I worked for the Land Institute for a few years when I first moved to Kansas as a postdoc, and they think about things like intercropping. So not having an entire field of your cereal, whatever that might be, wheat or corn, but intercropping that with a legume and maybe more than one crop. And those things can help eliminate those high accumulations of pathogens as well.
You have perennials living for 100 years that are herbaceous. So this is not a tree.
Mm-mm.
Why can some people not get a tomato plant to live a full season? What's the difference there? Is it the domestication of some of these species and improvements we've made over time to the plant to get our intended yield? Why are some vegetative or cash crops for humans so susceptible to disease when you have naturalized plants living 100 years? That just seems insane.
Yeah, I mean, in terms of domestication, I think we picked annuals because annuals tend to reproduce every year, right? They have to. If they're not, that's a major problem for an annual plant. And the things that they do when they're reproducing tend to be what we eat, right? So grass, corn, an annual that we selected, we chose it because we wanted something that would make kernels every year for us to eat. So that's a great thing. So we tend to focus on these weedier plant species that are making fruits almost immediately. And yeah, that might not be so great for us in the long run. But there are some groups thinking about shifting from annual plantings to more perennial plantings. The Land Institute is one of those. They work with this grain called kernza, which is a perennial grain. And you can plant it and it can sit there more like a prairie plant, not for hundreds of years, but many years. And you don't have to till it, you don't have to replant it, and it can continue to make grain season after season. So there's been some success with some of these grains. They're working with an oilseed crop and a legume. They've had some major success with perennializing rice in China. So there are some steps moving towards that direction. And the role of the microbiome in perennializing agriculture, I think, is going to be huge.
Yeah, and this ties a little bit into a study that you were referenced on once, looking at the reproductive potential, size, shoot allocation, and growth rates of weedy plants may result from reduced resource allocation to other aspects of plants' growth and defense. And you looked at domesticated sunflower cultivars versus these improved cultivars versus ancient landrace plants, and there were differences there. Can you talk a little bit about that, of how these improved cultivars have differ than more naturalized plants?
Sure. So the Kansas native sunflower looks way different than what you're thinking of when you think of sunflower. So it's not this massive thing that's 9 foot tall with this huge head full of seeds that it can't even hold up. It's like falling over. That's not what the natural sunflower looks like in Kansas. The Helianthus annus is this bushy plant with a ton of flower heads, hundreds of flower heads with teeny tiny seeds and teeny tiny flowers. So we took that plant and we changed it to be this massive single stalk thing with this huge head with fat seeds for us, because it's much easier to harvest that than going through a million little flowers with some machine. So we selected that. And in the environment we were selecting it tended to be these fields that were tilled every year that had very poor mycorrhizal communities, for example. And these plants, the ones that did well, learned to adapt to those fields with poor mycorrhizal community and a ton of nitrogen and phosphorus applications, right? So as we were selecting for our crops for the traits we want, big fat seeds, we were doing it in this environment that also selected for plants that were less dependent on microbes and really liked a lot of fertilizer. And that's what we have now.
So if we had gone back in time to when they started selecting and we started selecting in the native environment, could we have ended up in a result of the sunflower we know today, but it would have the longevity potential of the native species?
Yeah, I mean, maybe we could always go back and try things differently or try things again. I'm not sure. With the sunflowers that we eat tend to all be the annual variety for the most part. I don't know that there are perennial sunflowers or helianthus species, but I don't know that any of them have been domesticated. And if anybody is working on that, but that would surely be interesting.
As part of the food trade-off, right, is fertilizers relatively cheap.
Yeah.
Annual crops grow quick. They produce food relatively easily, relatively cheaply. So the whole idea of perennializing it, making it have microbial symbiosis and lengthening out the cycle, maybe it's like, why go through all that effort when we can just do it this way? It's fast, it's easy, it works. We know how to do it. You know,
Exactly. And that's why it has been done that way since the advent of the steel plow in the 1880s or whatever. It's fast, it's easy, and it works. So why do anything else, right? I guess the downside of that is, in some of these places, we've lost a foot of topsoil, and that took, eons to create. And that's a problem. We're having declining fertility with the loss of topsoil and the constant tillage, for example. And as we learn more things about the chemicals that we spray on our plants, both in terms of harming our own human health and fertilization, harming our biota in our area and beyond. Like if you think of algal blooms in the ocean, for example, maybe it isn't the best way. Maybe it helps your corn in your field, but for how long? Is it going to be a fertile field for your family when you pass it on to them? Or is there going to be 8 more inches of topsoil lost in that generation?
If I had a low tillage or no tillage system with relatively low fertility and a robust microbial community, say I use an inoculant like your MycoBloom, and I put it in the soil and I grow tomatoes in that, and I do that for 20 years, and I'm saving seeds and replanting the same species, do you think I'm getting something at the end of that 20 years that's more adapted to that soil and is potentially forming relationships with the microbial communities in that soil because that's the conditions I've saved seeds in?
Yeah, I think that sums it up pretty well with what's happened with modern breeding, that example. Yeah. I think you would be selecting for microbes and plants that like to live with those microbes in that soil. Mm-hmm. Yeah.
So we can almost like I don't want to say undomesticate, but we could move some of these domesticated
re-domesticate.
Species. Yeah, it's more like a regenerative agriculture system.
Mm-hmm. Yeah, I think that's totally possible. Unfortunately, it seems like most of the plant and seed breeding that we do does not occur in those environments and with those considerations.
So here's a tie-in.
So far.
You have your product, MycoBloom, and we can talk a little bit about that coming up. But if I'm planting crops that are grown, have been bred to be in this like fertilization system, organic or not, and I add a microbial inoculant to the soil, are the plants that I put in that going to benefit from that additive?
Yeah. It depends on your plant. If it's a mycorrhizal plant, for instance, that's something to consider. If it's a broccoli or a brassica, it's not going to benefit from any mycorrhizae at all. So it depends on your plant. My strategy with MycoBloom is I make my MycoBloom mix as diverse as possible. And so I'm getting ready to release a new product. I don't totally know its name yet, but I'm starting to go with max diversity. And in this product, it has 14 different species, 23 different isolates. So just a ton of fungal. The diversity is really high. And that's in contrast to some of the other inoculant producers, which might have one species or some smaller number of species. And my thought with that, and it's based on nature, is that if you give enough potential via diversity for these fungi to excel in the soil, something is going to work really well. So out of these 23 different isolates, somebody is going to be super great at acquiring the calcium from your soil. Some are going to like it when it's really wet. Some are going to like it when it's really dry. So no matter what the situation, that whole inocula can do well because some individuals in that inocula can perform well in that soil with that plant.
One question I had, since you're a scientist, you have this company, and I love that it's being led by scientists, not somebody just doing marketing and, taking advantage of a trend.
I also do the marketing. Yeah.
Okay. Oh,
I wear all the hats.
there you go. Me too. If you go on Amazon, you search, and I know you guys are on Amazon, it comes up, there's a ton.
So many. When I started, I think there was 4 pages, and now there's like 24 pages, just 5 years later. It's really blowing up the number of products out there.
How does somebody begin to even suss out A from B? Because the first thing people are going to look at is price.
Yeah.
And they're going to say, okay, what is the cost per pound, per ounce, whatever it is. And then there's probably a lot of jargon that you've seen that people put on stuff to make it sound maybe better than it is, or that doesn't, it's like the best coffee in the world or whatever.
High performing strains or whatever. Yeah.
Yeah. But what does that actually mean? Right. Or we have 50 different species, but if most of those species are in such low concentrations that it's ineffective, who cares? Like, you can say they're in there. But going back to the question, how do people begin to sort out products if they want to go down the road of microbial inoculants?
Yeah, I mean, I think probably what most people do is Google inoculants on Amazon and buy the first thing, right? There's not really a lot of thought, and I'm happy for those guys. They're doing well at the top of Amazon. I'm on like page 3 now. But one thing that I've always struggled with in the industry, and one of the reasons I made MycoBloom, first of all, there was no local Ecotype products, so nothing for prairie specific. So I wanted to meet that need. I also was just super underwhelmed in the experiments that we did with our lab working with these commercial inocula. Some of them actually were worse than the control trials that we had set up. So the plants did worse with these inocula. And in my career, looking at some of these inoculum, it's just super disappointing because they say things like we have 1,500 papules per gram, and then you actually look what's in that, and it's more like you have 2 papules per gram. Like, that's a crazy, crazy, crazy overestimate. And so it's unfortunate that the industry doesn't have tighter regulations. There's not really people looking at these labels in charge of saying you do have these 19 species or whatever at some concentration. So a lot of the stuff that people are selling isn't up to snuff. It's not what they say is on their label. So I think that is a major problem with the industry, and why some people become really frustrated when they buy a mycorrhizal product and it doesn't work, or it works for just a short period of time because it had, nitrogen and phosphorus in it, but that was a temporary thing, and then it doesn't work anymore. So that's really disappointing. And trying to find a product that can work and is viable and at least has something close to what it says it has is a challenge, right? Because you don't have a microscope and 14 years of experience working with, mycorrhizal fungi. It's not a question that many people can answer at home if their product is viable and is what it is. So that's a major challenge.
That's the thing. It's not, I can see it, I can't taste it, I can't smell it. I don't know. Like, I'm looking at dust or granules or something, and like, it could be dirt, or it could be the best inoculant in the world, and we don't have the
Yeah.
biological equipment to tell the difference. Is it a case of, again, most farmers are the people listening to this, of if people buy food, they connect with their farmer, they know their farmer. So I know how the food's being raised. I can go into any big box grocery store and I can go into an aisle and I see a picture of a farmhouse on something that we all know wasn't produced on a farm like that. And it's just the marketing of it. Where in a case like this, is it do your research and spend some time on some websites and go with your gut, buy from somebody you trust? Because if you just look at the bullet point features and benefits on Amazon, or some of the aggressive names, killer mycos or something like that, right? Like you
They
can
have
get,
some really fun names, some of these products.
they do. The whole
Yeah.
cannabis industry is, I'm jealous
Mm-hmm.
of what they can name
Yeah.
their products.
Mm-hmm. Yeah. And so it is challenging, right? And yeah, it is just such a challenge. What I try to tell people is look at the plant. So with my trials, I can grow a plant in a pot, my control, versus a plant in a pot with my inoculant that I'm testing, whether it be MycoBloom or if I'm doing, my academic isolates. I can see a response in that plant in as little as 3 weeks. So that's something you can visualize how the plant is responding to that fungi without seeing the fungi. So that is a possibility. And thinking on the larger term scale of farms, I did some work with a local tomato and pepper farmer in Lawrence, Kansas, the past couple of years, and a hemp farmer. And in these trials, we were noticing more tomatoes per plant and ended up having something like 1.5 pounds more tomatoes per plant on average across these sites. So there are some measurable differences. It does take a lot of data to get down to the pound of tomato per plant. Not every farmer is going to do that, and I absolutely understand. But when you're growing your tomato starts, you can see if a tomato is happy and not happy by looking at some of these products. And for some of these products, that could be that unreported, or sometimes they do report it, addition of nitrogen, for example, or phosphorus, whatever fertilizers they put in there. So that can be a transient thing that's not linked to the microbes. But yeah, it is really a challenge if you don't know what you're looking at.
So that's the other thing. I put it on, or I use it, and it's like, well, did it do anything? So without doing your own kind of, I think a lot of people to get comfortable on this or to scale it up, it's do a control, right?
Yeah.
These plants with this added, it's relatively inexpensive. You're
Mm-hmm.
$24 for, I think that's a 2-pound bag on Amazon. People could get it, so they can check it out. MycoBloom,
Mm-hmm.
MycoBloom, or mycobloom.com is the product. So I could do a trial on my own and then, compare it. Does the user potentially also run into the issue that we went back into of prairie restoration? What soil am I dealing with? So I could use the best myco product in the world, but if I'm putting that into a garden that's full of synthetic fertilizers, I might as well not even use it.
Yeah, so the farmer that I worked with at the tomato and pepper trial recently was an organic farmer, right? But he did every week, twice a week, fertilize the plants with, typical USDA approved organic fertilizer methods, pumping it through the irrigation or whatever. And even in this context, with the amount of materials he was supplying to those plants, the mycorrhizae did help, even with weekly, twice a week fertilizer. And I would say probably in most gardens, you're not dumping enough of anything to really kill these fungi in the short term. They might not survive for 10 or 15 years if you're constantly fertilizing the soil or tilling the soil, but they can survive a little while. And that brings up, well, then do we have to re-inoculate the soil in our farms to maintain these relationships or repair them? And if so, how often? Is it every season? Every 2 seasons, every 5 seasons? When do we need that refresher of these beneficial microbes if we're putting them in suboptimal conditions and we expect a decline, such as if we're fertilizing all the time or tilling all the time? Those questions aren't answered yet.
Say we're in a no-till system, and say it's a typical garden. I inoculate in my potting mix at the beginning of the year. I plant it out. It gets transferred into the soil. those microbes and plants thrive. They're doing that synergy. At the end of the season, crops go away. We can assume some spores have been formed. And I'll just say we'll use your location, Kansas, because you're, not too far north, you're not too far south. What would happen? Is there enough spores in the soil to remain there over the winter come spring to reform relationships?
Yeah, I mean, in the prairie, absolutely. They're just hiding out in these dormant long-term perennial roots, right? But if you're thinking about more of an annual ag system, um, yeah, there should be enough. If they go to spore form, they should be safe in the soil. And, mycorrhizae are adapted to soil and the things that happen in soil, even a little bit of freeze thaw. And so they will be able to survive a winter, sure. Yeah.
How local is local? So I'm in California, you're in Kansas, somebody listening to this is in Maine or Florida. I get a product like MycoBloom. Am I gonna, if we all planted tomato, are we all gonna get different performance because of our local climate?
Yeah, I would say for most products, that would be totally true because most products are one isolate and it's going to do different things in different places. So I tried to target this by creating this diverse mix so that something can interact with your California soils and something like the hummus in Maine, and so that this product can perform well in a number of different locations. But in terms of like the scientific assessment of how local is local for these AIM fungi, we've done some studies looking at fungi from Indiana and how they perform versus fungi from Illinois versus Kansas. And, these are places that are 10 hours away in a car. So pretty local, I'd say. And there are some differences in how these fungi can perform just at that short distance. But the community, if you cross transplant them, can do pretty well in both places.
Is it a symbiosis thing? Is because I talked to somebody else about this and I forget which interview it was, where they pulled out, say, strains out of their inoculant that they had for the soil. It was a biological. And they'd put one in with a plant and you didn't really see great results. And then you'd have like two species in with the plant and you got better results. And then once you added three, you got even better results. So it wasn't, you couldn't pinpoint it was like one specific strain that was making the difference. And none of them worked great with just one strain. It was only when you brought together this consortium of a bunch that you saw the results. Does that hold
Yeah,
up
I love that word
scientifically?
consortium. It's such a great word. But so with the AM fungi, there's been a ton of studies looking at individual assessments of a plant with an individual fungi and also with a mixture of all those fungi. And generally, if you do a meta-analysis, which is like a study looking at all the studies out there, compiling all that data and finding the average pattern. Generally, what happens with these mycorrhizae, it seems the best fungus as an individual performs about as well as that mixture. And that mixture is about the same level as that best individual fungus. But what the best individual fungus is will depend on a given soil type or a given plant, for example.
So
Does that make sense?
I have quantity 10 of A and I have quantity 10 of B, C, D, and E. They'll perform equal, but I have to make sure that that A is the best fit for whatever I'm growing.
I would say if A is up here and B is here and C is here, if you mix all of them together, you'll get basically A again.
Got it.
So it's not necessarily additive. It's whatever that best individual is, that's where that mixture is going to be basically. But A, B, and C for soybeans, for example, and maybe it's like this, A, B, and C for corn. And so, but similarly, this best fungus for corn is probably going to perform as well as that mixture for corn. That tends to be the pattern we're observing with these mycorrhizal fungi. So it's not necessarily additive. It's driven by the most beneficial species in the mixture, but who is going to be the most beneficial depends on the soil and the plant and all the other contexts.
If I have a thriving farmer garden and I add an inoculant to it, can I mess up what local population I have built there over time?
Yeah, I mean, if you have something that's thriving, I wouldn't mess with it at all. You're doing great. So that's something to consider. But there has been some work looking at can these inocula actually replace the fungi that are present in an area? And there is some evidence that it can happen, especially with commercial inocula. And I hate to, diss the industry because I'm also in it, but many of the species that are available in these commercial inoculants are actually very ruderal. They're very weed-like. They tend to grow really fast, reproduce really fast, have a ton of spores. And those traits that make them easy to culture and put in a product, similarly, if you put that in nature, they're going to grow and reproduce really quickly, and they can actually outcompete that native community in some cases. There's been some recent work that has found this to be true.
Is that a worry? People worry about invasive
Yeah.
plant species coming in. Is there
Yeah.
an invasive microbe threat potentially on the horizon? I know these things aren't
Possibly.
widespread, but yeah,
If it is going to be any species, it's going to be Rhizophagus enteratices, formerly known as Glomus enteratices, and now their names are changing all the time, as Rhizophagus aurelus. That species is in almost every commercial product, and many of these commercial products are shipped across the globe. So if there is going to be, that non-native fungus that is gone everywhere, it's definitely that species.
If I'm a gardener, a farmer, would you say the best way of getting this into the soil would be into my potting mix when I seed plants and then transplant that into the field? Is that the best way?
I'd say that works really, really well. So I often do this thing I call the inoculated nurse plant. So it's basically when you're growing your little plugs in the greenhouse with that small amount of soil, you put just a small amount of inocula with that plant, and you allow that inocula to grow up with that root system over time for a few weeks prior to transplanting. That's the best way to make sure that these mycorrhizae have a host that is going to feed them, because they're already associating with plants that are feeding them sugar, and they're doing well. They have that connection going on. They can survive. It's also a good way to put out small amounts of inocula, so you don't have to inoculate the whole landscape, just right where those plants go.
Say I follow that strategy and I do that with plant number 1. And then on either side, I don't inoculate those plants. Plant 1's inoculated, goes into the garden. Plant 2 and 3 haven't been inoculated in the nursery. Will the fungi working off plant 1, can 2 and 3 start forming relationships with those? Is there some transfer, you think?
They absolutely can travel via their hyphal connections. And, if as they're exploring the soil, they might meet a root of another plant, and absolutely they'll colonize that plant. And then, if that plant is good at feeding it, it'll go on and on. We've done some work in the lab in prairies. The spread of that source of inocula does happen, but it's pretty slow. It's about, I think it was 1.5 meters a year. So that's at the end of the growing season. All this inocula only traveled about 1.5 meters. So what is that? 4 or 5 feet. So that's something to consider. That would be a way of cutting down cost of inocula for sure.
It's still pretty far, given that it's so tiny.
Yeah, it's very tiny. And not all the fungi moved at the same space. Some were better at going than others, but generally there was some good movement at 5 feet after about a year.
Brassicas don't form relationships. Are there any other edibles or common garden plants that people would be wasting money on if they tried?
Not really in the garden. I mean, I guess blueberries, they associate with the Iroquoid fungi and some of those types of plants, and some of the tree plants that make nuts, I think, are they associate more with the ectomycorrhizal fungi, oaks, for example. But many of the vegetables that we eat and plant in our gardens associate with arbuscular mycorrhizal fungi. So your potatoes, your basil, your corn, your beans, all that stuff love mycorrhizae.
Fruit trees?
What was that?
Fruit trees, like apples,
fruit trees?
peas?
Yeah, apples associate with AMF.
Okay.
Yeah. I don't think about the trees too much. They grow too slowly, but I say that working in the prairie, they're very slow growing too. But
yeah, it's one of those things, so I've talked to a bunch of people on biochar, and there's, mixed results on that depending on how you view them. And I kind of view it at this point of like, hey, it's not really a negative. There might be some positive upside to it if you can control the costs, right? It all comes down to money. If you're spending a ton of money on biocharing your land, well, then it becomes a financial problem. And I look at a lot of myco inoculants like kind of the same way. I think,
Mhm.
right, like it's relatively cheap and there's no negative. You're probably going to get a bump from it. It's kind of insurance for the garden, right?
Yeah. I would say that's probably true. The risk of invasiveness of these inocula, well, first of all, it's probably already too late, right? There's a million products on Amazon and they're all over the world. That's, I guess, a very pessimistic view of it, but it's probably not going to hurt anything. That's true. Um, I try to tell people if they're going to buy something, try to think as local as you can. So there are products that are, produced in the U.S. MycoBloom definitely is produced in Lawrence, Kansas, and there are companies where their isolates are also, at least from a region nearby, and there's products whose isolates come from another continent. So that's something to consider, I think, when choosing a product. What relationship you want to have with that and the potential for invasion. Another thing I tell people is to think about granular products. Those, in my assessment, tend to be the ones that do have the highest spore counts, and they're often grown in actual media. We call it in vivo versus in vitro cultivation, which happens in plates with genetically modified roots where they essentially just scoop out the spores and put it in some powder or something. The in vivo grown, the ones with the granular, they tend to be more viable, it seems.
If I had a granulars to potting mix, do I need to use that potting mix right away? You mentioned earlier sometimes when they get wet, they can start to grow, and if they don't find a host.
Yeah, if you can put it in your mix and keep it dry, it'll be fine. But yeah, once you start to get it wet, I would try to put a plant in there within 2 weeks.
Does storage requirement and or expiration date have any relationship with quality on these products?
Yeah, so these fungi, when they're dry, so like my MycoBloom is this granular thing that's super dry, and then I put it in a package. So in their dry form, some of my spores have lived for more than 6 years in that package. So I go back and periodically test it. So that's a really long time. Once you get it wet, it's a whole nother story. Other microbes will start eating those fungi and they can start to germinate the spores of the AMF and die. So once it's wet, it's a totally different story. Another thing to think about is that they're alive. And like most living things, they don't like to be too hot. So they start to die around 140 F. So for example, if I have my little MycoBloom package, the sample I brought in today, if I were to leave this in my car, it's probably dead. So that is something to consider where you store it. In terms of cold, that's not as much of an issue. Many of these fungi are adapted to freeze-thaw cycles. It's just the heat, they get killed right about, it's just slightly hotter than humans.
What are your thoughts on cultivating AMF from home, not from a culture like yours, but from something like native in the soil? David Dowd's, I think USDA published some information on this a while back I think he was taking something like planting grasses in bags, inoculating those, growing it out, and then you'd have the AMF in that soil, and you could use that soil to then inoculate other areas. Is that is it possible for people to clone, whatever the word is, reproduce local AMF?
Yeah, with certain considerations. So for example, in the prairie, we do whole soil transplants for restoration. The downside of that is you have to dig up an old growth prairie to collect that soil with that awesome mycorrhizae. So that's something to consider. And in some of the places I've worked, for example, in Indiana, if we're doing a prairie restoration there and there's only 5 acres of prairie basically left in the state, like they're not going to allow you to dig up any of those microbes for sure. So even access can be a challenge. But if you have a land that you think has good beneficial mycorrhizae, it is possible to cultivate it. You do have to consider, if you're going to use it as an inocula, that it is a living thing and it has a life cycle and a response and a requirement of a host plant. So you have to make sure you're picking a good host to feed that fungi. If you want to propagate it, you have to basically kill that plant and allow that fungi to go in spore form before you start your next round. So it is totally possible. it does take some time. So the way we culture our fungi, it requires about 6 to 7 months of growing before we start to allow that plant to senesce. So it definitely takes some time.
So it is doable if somebody was on it
It's
real
doable.
DIY. Hey, I want to do
Yeah.
this
Hmm.
in-house type thing.
The benefit of just looking at particular species such as mycorrhizal inoculant versus a whole soil is in that whole soil, you can also culture other things that aren't mycorrhizae. For example, pathogens, nematodes, other microbiota that's in that soil. So that's something you have to keep an eye on for sure. So you're not putting pathogens and root rot fungus everywhere, for example.
Is there a carrying capacity or upper limit for AMF? Like say money was no object and I put an inch of MycoBloom across the top of the
Yeah.
soil. Am I going to get better performance than if I put, you know, a millionth of a percent less?
Yeah. So I tried to get at this I think it was my restoration ecology paper that came out in 2022, which is unfortunately behind a paywall. So I don't blame you if you haven't read it yet. But in this study, it was an inoculation density gradient. So we had these plots where we added no fungi, like the smallest amount of dust, like 2 kilograms per hectare, to what I call dump truck loads. So massive amounts of soil. You could literally see it sitting, millimeters thick on the soil surface, massive amounts of inocula. And for the most part, plants responded somewhere in between. But there were some plants, in particular our native legumes, didn't have an upper limit. So even with that dump truck loads per acre, we were still seeing benefits with more inocula, indicating if we were to put double dump truck loads on the next acre, we would see double the legumes. So it really did kind of depend. But most plants were somewhere in that middle range. They found the fungi that they preferred and kind of leveled off.
What was the response to more? Was it like what was the plant? What did the plant show that was saying I like more? Was it just bigger? What was it?
Yeah, so in this study, we were looking at the above ground biomass of the plants. And so we would go through and we do this thing called point intercept method. We're getting real in the weeds now, but basically just looking at the abundance of the plants that are there. So not only did we see more of these native legume species, but the ones that are there were doing really well versus in some of the plots with lower inocula, that plant might be there, but it's still really struggling, even though it was the fourth year of the restoration. They're just tiny and who knows if they're going to make it.
Can any of the species help aid the defense of a plant, either trigger something genetically within the plant, by giving the plant additional minerals from the soil that it otherwise couldn't accumulate, like it can defend itself better? Do you get a plant defense bump from
Yeah,
AMF?
absolutely. Especially when thinking about those little seedlings that we often grow up in, our still potting soil in our very protected greenhouse. Sometimes when those are transplant, that's why transplant shock happens, right? These plants have not been triggered to have a defense system to be able to handle any wind or drought or all the conditions of the world. Fungi can trigger these defensive pathways and help prime that plant to be able to deal with other when an actual thing that comes to harm them comes along. Another example, I did some work with milkweeds in the prairie system and looked at the amount of latex produced by these milkweeds. So if you know the story of milkweeds, they're hosts for the monarch butterflies, which I don't know if they're federally endangered or if they just didn't make the cut this year. I know it was a hot topic in the news. But these monarch butterflies are obligately dependent on the milkweeds because they eat the sap of the milkweeds, they internalize that latex and the cardenolides, and then they're defended and they can persist. And so in the study, I showed that if you gave these milkweeds our native mycorrhizae, you can trigger this plant to actually make more latex. And so these plants were healthier, and even plants that were the same size had more latex with the fungi than without. And so, from the perspective of the plant, it's better defended against most things because most things don't like latex. But from the unique perspective of the monarch butterfly, which loves latex and cardenolides, having more of that makes them better defended as well.
Yeah, I guess I'm thinking, right? Like if AMF are primarily bringing more phosphorus to the plant. Anything that plant would use phosphorus for is now dialed up, right? Because now
Yeah.
I have more of it.
Yep. Sure. It's basically no longer limiting if you have fungi that are working hard. They'll get you as much phosphorus as you want because, carbon is not a limiting thing for plants. They just pick it up, photosynthesize, drop it out their root. So they can feed fungi if there's something that they deliver.
What's the holy grail right now in AMF research? Like when we'd love it if we could get AMF to do this.
I don't know. There's so many different dimensions that people go. There's a ton of scientists, and it's a really growing field. AMF ecology, systematics, taxonomy, application. There's just so many things. I think what's really interesting about these fungi is that they are so genetically complicated. We don't actually even know what's out there. Even though they're micro, we can see them in the microscope during microscopy. But yet we've only described around 345 or 46 species of them. And there's probably orders of magnitude more. So even knowing what's there, I think can be really important. And we won't know fully how the things we do to our land or how our inoculants work until we actually do know what is out there. And that's a growing field for sure.
There's this like theory that plants farm us, not vice versa. Like we've spread, potatoes and all these plants all around the world, and they're thriving because, hey, we like them, but they're the ones surviving.
Mm-hmm.
Are fungi farming plants?
Mm-hmm. Yeah, maybe. I mean, maybe, yeah, I could see a fungus giving more phosphorus to a plant that's giving it more carbon for sure.
Namely, what
Yeah.
if you had to go out? I mean, this is way out there, sci-fi-esque. Knowing what you know, I mean, within the realm of reality, like, is there things that you think could be possible in, say, our lifetime with AMF plant relationships? Like, we don't know it now, or we don't know how to harness it now, but are there things that we think could be there?
Hmm. I don't know.
Yeah, it's a wild question.
Yeah, I don't
I
know.
don't either. If you had to sum this up, if we know how much about AMF from 0 to 100%.
Yeah, they're cryptic. I would say 41%. That's my guess.
So less than
hmm
half. Like there's just
Less
still a massive
there's
amount of
just a ton
unknown.
we don't know. Even like if you look at the commercial products and they don't even necessarily, it seems based on my assessment of them, even though I'm in this industry, like know what they're putting in there. They can't necessarily confirm it. I think that's astonishing. Yeah. How to get these things to grow consistently. It's way more challenging than breeding, let's say corn, a thing that you can see and touch. And, it wilts in front of you when it's not happy. So growing and cultivating these fungi and understanding them is just way more challenging.
Here's a question I ask everybody regarding their own field. Is there one piece of knowledge that's currently unknown that if you knew the answer to that, it would change your fundamental understanding of it or how it works? Is there something like one piece of mystery around AMF that, like, if you could just find that, it's like, whoa, okay, now this opens up a whole new thing?
Mhm. I I don't know that I have a particular direction for that question, but what I will say I think is the most mysterious about these fungi is, I mean, they're just strange in many ways, right? So their spores, for example, is a single cell, but it can have hundreds of nuclei in that cell. And furthermore, it's heterokaryotic, which means those nuclei can be genetically distinct from each other. And across a fungal's body, it can segregate these nuclei in different ways. So on this end of the hyphae, they're all type A. On this of the end of the hyphae, they're all type B. If you were to sample the soil here versus here, it would look like different individuals, and yet it's the same entity. And that is just insane to me that can happen in nature, that something can be so distinct. And also the species concept is a little bit strange because these hyphae confuse and pass nuclei across individuals. There's been some work where they might pass nuclei across species. And so then what is even a species? There's a lot we don't know about these fungi, and they're very strange and act differently than many organisms on Earth.
Do we know if they communicate amongst themselves, like if they are connecting?
Yeah, they must. I guess I'm not an expert in that realm. But yeah,
If
you would think so.
so, does every spore have the potential to be something totally different from what it
Yeah,
originally?
if you look at one so if I start a culture from a single spore and allow that to grow up for a year and it makes a million spores in a pot, I can sample those spores and they can be very different from each other,
You have a
which is
million
another
different things.
reason that when you put these fungi in nature, they're even within an individual, they're so genetically diverse that they can do a lot of different things.
I guess that's the whole
Because
benefit, right? That's why they're still around. It's just like
mm-hmm,
we have a million rolls of the dice here to
yep,
find the
something's
situation.
probably going to work.
Yeah.
Mm-hmm.
Well, that's
Exactly.
amazing. Thanks for venturing off into the weeds with me. For people that want to
Sure.
learn more about your company, they can go to mycobloom.com. They can read all about it. I think you've done a really good job on the site of explaining, what's different about this inoculant and how you go about it. So it's pretty transparent. I love that. For people that want to follow along, say on your Instagram, which you mentioned before, where can they go?
My Instagram is at MycoBloom. I think my Twitter as well. I just signed up for something called Threads, but I don't understand it yet.
All right, so check them out. Thanks so much.
Sure.